they will come to know that how this pharmacokinetic modeling can be used for designing different doses form, and what parameters, what will be the methods we should use for determining various pharmacokinetic parameters. Okay, so well, Nilesh, can you minimize this? Yes, it is okay. Right. So for the development of any dosage form, it's a long process, you know. It is starting from synthesis of new molecule to the designing of particular dosage form for getting any therapeutic effect. And the total time estimated for getting a one successful dosage form of any new drug, it requires at least say 13 to 14 years of exhaustive research on that drug and formulation of the particular dosage form. And it includes the preclinical studies which we perform after, say, synthesizing different compounds. Say if you synthesize 500 or 600, 5,000 or 6,000 compounds, there may be only 2 or 3 compounds, right, which may be active, and then we take these compounds which are active one, which are showing good physicochemical property, and we thought that these will be the good for getting the particular response. Then we'll test these compounds on the animals. That will be the preclinical studies. Then we'll switch over, say, if we get the successful preclinical studies, means experiment that we perform on the animal. If we get the successful result, then we switch it over to the clinical trials. That you know that there are 3 clinical trials. Clinical trials 1, phase 1, phase 2, phase 3, where we assess the compound for its safety and efficacy, right? And then we'll switch over to the say if we get successful phase 3 clinical trial, then all these data will submit to the drug department for marketing that product to the marketing that product. So all the data will submit to drug department or FDA department as a new drug application. Then Food and Drug Administration department review your data. If they find that yes, you are reliable and good, it is not having any serious adverse effect on the human being, then they will allow you to market it, but they will allow you to market for certain area, not for whole area, for whole country. You have to define the area, right? So in the limited area, you can market that product, but it will be responsibility of the manufacturer to get feedback from the market. That is called post-surveillance studies. That is the phase 4 studies, clinical phase 4 studies. So under this clinical 4 phase studies, if no adverse reaction observed during the 3 or 4 years of its marketing, then again we have to approach drug department to open that market so that you can market that product to the whole country. That will expand the area of the marketing. So this is how the drug or dosage form will be developed, and how the dosage form will come from the lab to the market. It's a really tedious process. It is a time-consuming one. It is expensive also, right? So what happens, say, after getting the successful product, in between also you will require to study different pharmacokinetic studies, right, of the product. And say if you give same means if a drug is manufactured by say 3, 4, 5 manufacturers, their therapeutic level means their drug level in the body after its administration will be different, right? So if the different formulation will be different, naturally their level in the body will be different one. The drug level in the body will be different one. Similarly, if the same drug is administered through the same route, right, same route in different doses form, naturally if the different doses form is not different doses, not doses form in different doses, means different concentration. If you administer different concentration, the level of the, say if the doses form is having a low concentration, then this type will graph you will get. If you further increase the drug concentration, then the drug concentration in the body will be increasing. So as on increasing the concentration of the drug in the body, the concentration of the as on increasing the concentration of the drug in the dosage form, the after the administration, the concentration in the body will be increasing one. And for getting the response of the drug has to cross this minimum effective concentration. If the drug concentration in the body is not crossing the minimum effective concentration, it will not show any response, right? So you have to optimize the concentration of drug in the dosage form so that it will keep drug concentration in the above the minimum effective concentration and maximum the safe level, right? So this is the intention of the pharmacist, to keep drug concentration in this therapeutic window. Therapeutic window means that is the above the minimum effective concentration and lower the maximum safe level. Now, the route of administration, there are different routes of the administration by which a drug will be administered to the body for getting the particular response. Different, if the same drug is administered through the different routes, the different plasma concentration you will get, or different concentration of the drug in the body you will have. Say if the drug is administered through the intravenous administration, means the drug is directly administered to the blood, right? So at the administration time, that is the time 0, the concentration of the drug will be highest one in the body, means because the whole amount will be reaching to the blood, right? And then because of distribution, elimination, and metabolism, the drug concentration in the body will be gradually decreasing, right? Similarly, if the drug concentration if the drug is administered through the intramuscular route, then you will get there is a slowly increase in the drug concentration in the blood or in the body because of absorption of the drug from the administration side. Same is you will find in the subcutaneous absorption. So in this both administration, intramuscular or subcutaneous, there is a gradual increase. There will gradually decrease in the drug concentration. So what happened? The peak level will achieve. That we'll discuss in the next slide. In case of the oral absorption, drug if it is given through the oral route, it will go into the intestine, gastrointestinal and lumen, and from there it will dissolve and then it will absorb it reach to the blood circulation. So it will be taking in the time. So if you see the peak concentration is the maximum concentration that is achieving after administering the drug to different routes. In this case, the peak concentration is almost 0 time at the time of the administration in case of the intravenous administration. But in case of the intramuscular, the peak concentration is reaching at this time. Similarly, in case of the subcutaneous route, there is a delay in the achieving the peak concentration because delay in the absorption of the drug. Similarly, in case of the oral administration, there is a delay in the long delay in the achieving the peak concentration, right? And the dosing of the drug, the dose which we decide in any formulation, it depends on the rate of the absorption of the drug and concentration of the drug in the plasma, that is really the effective plasma concentration, and the rate of drug elimination. The absorption and elimination of the drug will decide how much of the drug will remain in the plasma at particular time period. So by these 3 parameters will decide how much dose to be administered for getting the response of the drug. Similarly, the pharmacokinetics help in optimizing the drug therapy, like doses, what dose to be delivered, what doses regimen. Doses regimens means the frequency of the drug that to be delivered to any patient or any human volunteer for getting the response for whole day, say 3 times a day or 2 times a day. That is the frequency of the dosing. That is called doses regimen. Another is the dosage form. In which dosage form it should be administered so that you will get the response of the drug, means the fast onset of action and long duration of action. So for getting the नीलेश ये लाइन आ रहा है कुछ,
ये आपकी
स्क्रीन पे लाइन आ रहा है ना, ये मेरे पीपीटी से ही है, थोड़ा मैं मिनिमाइज कर रहा हूँ
जी, कोई दिक्कत नहीं,
इसको मिनिमाइज करके मैं इसको अलग करता हूँ
जी जी।
चलने दे, should we continue with these lines?
हाँ सर, कंटिन्यू, कोई दिक्कत नहीं, करने दीजिए,
because I'm not able to remove these lines. How it comes out? चलने दे,
हाँ सर, कोई दिक्कत नहीं, चलने दीजिए।
Okay, so well, for developing any dosage form, for what we develop any dosage form? Just for getting the desired effect of any drug. Okay, so for getting any desired effect, we will require a right drug that to be delivered to right tissues at right concentration. Right concentration means the minimum effective concentration for getting the response of the drug. So the right drug to be delivered to the right tissue. Right tissue means where the action is desired. It to be delivered to the right tissues at right concentration means the minimum effective concentration for right amount of the time. Right time. It should not be like this. After the administration of the drug, you want a response immediately, but you are getting the response after say 2 hours or 3 hours. Then it will not have any use. Right? So question is, do we have a right drug? That we have to think. For getting the desired therapeutic effect, do we have right drug? If we do have, then how do we appropriately administer? Because this route of administration will decide how much of the drug should reach to the, desired tissues. आप लोग से आप अपने माइक म्यूट कर दें प्लीज. So after the administration of the drug, what will be its pharmacokinetics? Actually, what is pharmacokinetics? If we ask any layman, what is the pharmacokinetics? It is a pharmaco and kinetics, right? Pharmaco means it is the kinetics of ADME, right? ADME means absorption, distribution, metabolism, and elimination. So after the administration of the drug, right drug, after the administration of the right drug through the appropriate route of administration, what will be its absorption? What will be its distribution? What will be its metabolism? And what will be its elimination? Because all these all four parameters ADME will decide what concentration should remain to the right tissues, right, at particular time period. So you must have to know in that way the pharmacokinetic state. So to produce the desired drug concentration, what is the goal of the pharmacokineticians? To produce a desired drug concentration in the body using the optimal doses regimen and dosage form, right? Say the main interest is the desired concentration should reach to the target organ where the action is desired, and what up to what sorry and up to what time you will get the response, right? So that depends on the doses regimen. Okay, so what is the desired drug concentration? That you have to find out. What will be the desired concentration? That will be determined by the pharmacokinetic modeling, different pharmacokinetic modeling, right? This is the drug dynamics in the body. Say that dynamics in the body means the movement of the drug in the body. That is the dynamics, right? So it is the drug dynamics in the body. When we administer suitable dose in the suitable doses form to the body through the appropriate route of administration, whether it is intravenous or oral, it will reach, first of all, it will reach into the blood, right, or plasma, right? So the drug, after the administration of the drug will get absorbed. If it is not through the intravenous route, in the case of the intravenous route, drug is directly administered to the blood, right? There is no absorption. But in case of the other than oral route, other than intravenous route, drug has to absorb first, then it will reach to the blood. Whatever route of administration you adopt, drug first has to reach into the blood. Blood is the carrier for carrying the drug to the particular organ where the action is desired, right? So after the reaching the drug in the blood, it may bound to the blood protein, and part of the drug may bound to the protein, part of the drug may be free in the blood, and the free drug will be distributed in different part of the body, right? And as soon as drug will reach into the blood, it will be distributed, it will be metabolized, it will be eliminated from the body. And all these four parameters, that is the ADME, absorption, distribution, metabolism, and elimination, will decide how much drug will remain into the body.
Excuse me, sir. Sorry to interrupt you. All participants are requested to,
okay.
If video is closed, there is a time running out in 10 minutes. So you can again join by clicking the same link and join the session. Thank you, sir.
So after 10 minutes.
After 10 minutes.
Okay, okay. But a bit 8 minutes left, na?
Please continue, sir.
हाँ, 8 minutes left, na?
अभी 10 हाँ 8 मिनट्स हैं अभी सर आप चलिए
हाँ,
continue, sir.
okay. So well, it's that after reaching the drug in the plasma, it will be distributed into different part of the body. Part of the drug will be metabolized, and part of the drug will be eliminated from the body, right? And all these metabolism, elimination, and distribution will decide how much drug will remain into the body. So this is the after the administration of the drug, this will concentration of the drug will be in the body, right? This is an example of oral administration or an example of the first order absorption. First order absorption means drug has to cross a biological membrane before reaching to the blood circulation, right? So it will reach into the peak level. At this level, the input rate, sorry, input rate will be equal to the output rate of the body. So this is called steady state or Cmax, right? So this drug concentration that should remain into this therapeutic window. The role of pharmacist is to keep drug concentration in this therapeutic window that is above the MEC and below the MSC, means maximum safe concentration. If it is exceeding this maximum safe level, you will get some side effect of the drug along with the beneficial effect of the drug. So your intention is to keep drug concentration in this therapeutic window, right? There are, as I told you, there are four processes after the administration of the drug, that is the absorption, distribution, metabolism, and elimination. So what happened after the administration of the drug? First, drug will be delivered to the site of administration, right? Then from there it will absorb, it will reach into the blood. As I told you, whatever route of administration you use, drug will first go into the plasma or blood, right? Blood will be the carrier for the drug to carry it to the particular organ, right? It has to reach until unless drug is not designed in such a way that it will deliver to the target organ, directly to the target organ. If you are using some conventional route of administration, conventional dosage form, then drug will reach into the first it reaches into the plasma, and from there it will be distributed, right? Distributed in different part of the body. Part of the drug will come back to the, after the distribution the plasma, part of the drug will metabolize and eliminated from the body. So after the this is the sojourn of the drug in the body, means journey of the drug in the body. After the administration of the drug into the body, what will happen to the drug? How the drug will be moving, how much drug will reach to the liver, how much drug will reach to the liver, how much drug reach to the kidney, how much drug reach to this arm or brain or legs, right, or intestine, how much drug it will reach. That will be decided by different rate constants. That is the absorption rate constant, distribution rate constant, and metabolism rate constant, and elimination rate constant. There are different rate constants. These are the different rate constants: 1, 2, 3, 4. Absorption, distribution, metabolism, elimination rate constant. That will decide how much drug will reach to particular part of the body for getting the response of the drug. So pharmacokinetics, as I told you, it is the kinetics of absorption, distribution, metabolism, and elimination. Pharmacokinetics is either directly or indirectly associated with just about every part of pharmaceutical business. Why? Say, in the research for getting the suitable molecules, for the treatment of particular ailment, knowing about the pharmacokinetics is very important. And for the development of particular dosage form, knowing pharmacokinetics is very important uh, uh, for fixing the dosage regimen, how much times it should be giving to the particular patient in a day, so that you will get the drug concentration in the therapeutic window for 24 hours, and you will get the response of the drug for 24 hours. Then toxicology and safety assessment of the drug and formulations, knowing pharmacokinetic parameters is very important. Similarly, dosing recommendation for the age group or subpopulations, right? Say those who are having renal failure, hepatic disease, or rhesus patient. In that case, what dose to be administered for getting the response, because in these cases the dose adjustment is very important. That can only be done when you know different pharmacokinetic parameters of the drug. Then the effect of the meal and dosing, that is also very important. Say, as you might have a study about the various factors which are affecting the absorption of the drug from the gastrointestinal tract. There are biological factors, there are physicochemical factor or dosage form related factors. So meal is one of the biological factors that will affect the absorption of the drug, because some of the drug may bind with the components of the GIT and they may not absorb. So in case of the, can you ever think about the why vitamins are given after the meal? आपने कभी सोचा होगा कि why vitamins are given after the खाने के बाद ही विटामिन की जो बी कैप्सूल, डॉक्टर कहते हैं ना कि you have to take बी कैप्सूल after the meal, right? Because for complete absorption of the vitamins, you have to give it after the meal. आपको मालूम होगा, you might be knowing about the vitamins are absorbed by the active absorption process, active transport mechanism. Active transport mechanism, there are certain carriers are present in the GIT, and though these carriers will take the vitamins molecule and transfer it into the blood circulation, the serosal side, right? So if and there are limited number of carriers are present in the GIT, so that if the limited number of vitamin molecule will reach to the absorption side where the carriers are present for taking these vitamin molecules, if these carriers are not saturated with the vitamin, then whole vitamin molecules which are reaching to the site of absorption will be absorbed. But what happens if it is more molecules will reach to the absorption site where sufficient carriers are not present? Then there will be wastage of the vitamin molecules. The complete molecules of the vitamins will not absorb. So here the meal will decide. If we take after the meal, means the gastric emptying will be slow. खाने के बाद gastric emptying slow हो जाता है for complete digestion of the food. So if the gastric emptying is slow, then a small amount of the vitamin will be reaching to the small intestine where carriers are present for taking the vitamin molecules. So slowly the vitamins will be transferred from stomach to small intestine.